Randy, you don't mind being recorded, right?
No, I signed
Okay,
the
perfect. So
paper.
Randy is professor of epigenetics in the Department of Biological Sciences at North Carolina State University, and until recently, a senior visiting scientist at McCardle Laboratory here at UW. Randy was actually born in Kewaunee, Wisconsin, got his bachelor's, master's, and PhD degrees in nuclear engineering and radiation biology from UW, then spent much of his career at Duke University. He has done a lot to advance science, particularly in radiation exposure, and work that led to the identification of the IGFR2 receptor as a human tumor suppressor gene. However, his most significant accomplishment was showing how environmental agents can change mammalian DNA methylation and affect disease susceptibility in the offspring. This is his work on the agouti mouse, which he will tell us about today. Randy has received many national and international awards for his work, including the Linus Pauling Award from the Institute of Functional Medicine in 2014. He was nominated by the director of the National Institute of Drug Abuse to be Time Magazine's Person of the Year in 2007, was one of the main scientists featured on the PBS Nova special on epigenetics, which our prior resident Nirav Patel presented at these grand rounds a few years ago, and which was a main driver for our work in the lab. Finally, when presenting him with the Alexander Hollander Award in 2019, the Environmental Mutagenesis and Genomics Society stated that Randy's discovery represented one of the most important scientific advances of the 21st century. So Randy, I very much appreciate your being generous with your time. I am pretty sure that this is the first time you're speaking to clinicians, no less surgeons. You will find us different, and may have to explain certain terms for us, but I think you will find us both interesting and interested in what you have to say. I'm especially keen on you addressing our young residents and students because they are young and thus epigenetically much more modifiable than the faculty, so they can learn. There are others on the call, including, I believe John Deneau is on, who's the head of our epigenetics team at UW, Ruth
Right.
Kalish and Kirk Hogan that you know, Raghuva Moganti, who's an outstanding epigenetic researcher in our department, and others from different departments across campus. So sometimes it's about the big picture instead of the details of scientific experiment. So in light of this, Randy and I decided this will be an interview-based presentation. It's very different from what we usually do with grand rounds. We hope that you will find it interesting. So Randy, I'll guide you with some questions and then you take it from there. Most of the questions will discuss professional accomplishments because you've changed our understanding of the field of genetics. But I hope we'll have time for some personal thoughts because I suspect that you swam against the waves and defied common knowledge. Scientists rarely make big discoveries without going against the grain, and I'd like to know how you
As
survived
you
this.
know, Benny.
Yeah, I'd like to know how you survived this. I think we all would like to. So we'll go ahead and get started. Uh, so maybe we can start.
Before we get started, I want
Go
to thank
ahead.
you for that great introduction, and also for being willing to try this way of getting information across, because it's really interesting. I've done a lot of talks and actually, frankly, quite a few Zoom talks. And to me, Zoom is fine because you can get all over the world quickly. But the real problem with it, from my standpoint, is when you're giving an actual, like you would give a talk in a meeting hall personally, is there's absolutely no feedback. And it's very unsatisfying from the standpoint of the presenter. And when about 3 or 4 years ago, I was asked to do something like you asked me to do at Stanford University in a nutrition course. And we decided ultimately to go with the interview process. And I found it very, very, I liked it a lot because it's more relaxed and it's more informal. And in fact, it allowed with students, for example, even to get involved in the discussion rather than it being so just present the information and go away, and then try to figure out whether or not that went very well.
Yeah.
So that's why I suggested we do this. I know that this is not normally what's done for grand rounds, but Zoom, or this is WebEx, provides a mechanism by which we can do something a little different that's never ever done really in grand rounds, which is what we're hopefully will be doing today. So
Yeah.
that's my introduction.
Yeah, and we'll open it up for questions. I mean, hopefully I won't be the only one asking questions here. But let's start with just a brief your definition of epigenetics. What is epigenetics?
Well, epigenetics, to me, I define it differently through analogies. I think of epigenetics like this. I think of the genome, the DNA, as being comparable to the hardware of the computer, and therefore the epigenome, which just means above the genome, is the programs that tell that hardware or computer when, where, and how to work. So in effect, I think of a cell as being a programmable computer. And I think that is because I came through nuclear engineering and computer science. But I think the analogy works very, very well. There's two things going on in the cell. There's the genomics, the DNA, and in the computer, it'd be the hardware. And then there's the programs. And you can screw the computer or the cell up or make it even better sometimes by putting new chips or you're changing the hardware, but you can also do it through the software. And really, up until what you were talking about, our agouti mouse experiment, which was done in 2003, basically all the emphasis was on the hardware of the computer, missing, I think, actually a majority of the issues, which is the changing of the programs and what effects that then has on disease susceptibility.
Okay. Well, my second question was just from the standpoint of definitions. You've done a lot in imprinting, and I think imprinting is important to understand when someone needs to understand epigenetics. Could you please define this for us? Tell us about the protective effects of imprinting that you see and your hopes for imprintomics. You and I discussed this before, and I'd like you to maybe state it out for us.
It's interesting because usually when people I'm interviewed and stuff, we never really get into that at all because it's a subject that's a bit more difficult to understand and maybe even explain, but it's really, really important. In fact, back when I got into the field of epigenetics, which was back in the around 1995, really, and it's amazing how many years now that's been. But genomic imprinting was basically almost synonymous with the field of epigenetics at that time. There were two areas that people were working on at that time. That was DNA promoter methylation in cancer formation. That was just starting back then. And the field of epigenetics started back in the late 1980s, early 1990s. So what genomic imprinting is this: you inherit two copies of a gene, one from the mother and one from the father. And normally when those genes are expressed, I'm sorry, they're mowing my lawn outside, so I hope that's not, can you hear that noise?
That's fine.
Okay. Normally when the genes are expressed, both copies work. So there's a backup basically. If one is mutated, usually the other one will be able to work fine and everything is fine in the cell. But there's a subset of genes, and it looks like it's going to be around, my guess is people used to say 500, but I think it's more. It's going to be around 1,000 or so genes in our genome that even though we inherit one copy from the mom and one from the dad, these are autosomal genes now. But they're also evidence now we have that they're imprinted genes even on the X chromosome. With imprinted genes, one of the copies doesn't work. And that copy that doesn't work is silenced epigenetically. So now you've only got a single copy that works. So in effect, at all these loci, you're functionally haploid. So now a single mutation or a single epigenetic event, because this copy is regulated epigenetically, which means that it can inappropriately be overexpressed, or normally it could be underexpressed epigenetically, or it could be mutated. So you have no backup on these genes. And these genes tend to be growth regulatory genes. And they're also, interestingly, expressed in a parent of origin dependent manner. So IGF-2, for example, in us, when it's expressed, is expressed from, usually, it's expressed from the father's copy. Whereas the IGF-2 receptor, which is also imprinted but is an inhibitor of growth, paternal imprinted genes or expressed genes tend to be pro-growth. Maternally imprinted genes tend to be anti-growth. So in effect, you have a gas brake system in this whole thing. Uh, that's only expressed from the maternal allele. So you have these yin and yang genes, but they're off growth regulatory, so they're involved in development, they're involved in cancer formation, and there's only a single copy, so they're all susceptibility loci, extreme susceptibility loci. Because when you knock out one copy, you're increasing the probability of a mutation occurring by almost a millionfold, causing a phenotypic effect.
So what's the evolutionary advantage of imprinting?
The rationale for why this occurs is that it was developed and evolved because of a conflict between males and females to basically determine the nutrition that the offspring extracts from the mother, with the father supposedly trying to maximize that extraction to grow bigger and outcompete other embryos, even that might be sired by a different father, and the mother trying to dampen this down because she's related to all of her offspring. And if it grows too big in utero, the mother will die in utero. So it's a very fine balance between the maternal and paternally imprinted genes, and it's literally a genetic war, according to the conflict theory. So,
Okay.
and it gives rise now to susceptibility loci that are involved in cancer formation and behavioral issues. So, in effect, now these things are the cost of doing evolution.
Mhm. So aside
It's
from
really
imprint,
amazing.
yeah, yeah. So aside from imprinting, we know that some genes are more susceptible to environmental manipulations than others. What's your thought on this? Why is that, and how do we understand this better?
Well, there are a lot, epigenetics, and the reason why we have 260 different cell types is because of epigenetics. I mean, initially, the DNA is the same in every cell, but you've got 260 different cell types, and those develop embryonically because they literally, during embryogenesis, set up different programs in the cell. So each cell is, in effect, running a different computer program. So it's like you set up 260 different computers, and one's a browser, one's running Word, one's running Excel. That would be a liver cell, an eye cell, a brain, a neuron, and etc. So there's a lot of epigenetic programming that occurs early on that's not involving, imprint regulation or these metastable epialleles, which is what the agouti locus is in this mouse model that we'll talk about later on. So there are two really, I call them epigenetically labile um, groups of genes within the genome, and one is the imprinted genes, and the other are the metastable epialleles. The metastable epialleles, now that group of genes has been mapped by Rob Waterland probably 2 or 3 years ago in humans. So there's a map of these already in humans. They're present in us too, not just in mice. But it's not the agouti gene in us. There are other genes that are regulated this way.
Mhm.
And then you have imprinted genes, which we're literally at the end stage now writing this up. And there's, I think, 1,496 putative imprint regulatory elements that we have defined now. And of those, about 282, we have good enough data from sperm and egg to show that they're parent of origin to dependent methylation. So that we call the human imprintome. It's not the repertoire of genes that are imprinted, but it's the repertoire of the imprint regulatory elements that control the imprinting of these genes. That's now done. And it's really astonishing when you start now looking at that and map it into regions, which we've done, like Down syndrome, critical. There's 4 imprinted regulatory elements in the Down critical region. Yeah, not known before. They're there. The same thing with the George syndrome. There's about, I think, 3 or 4 in there. So there are imprinted genes that are in areas that we don't even know about right now because we didn't have this information that involve imprint deregulation in the formation and etiology of these disorders.
So I want to be careful not to overwhelm the audience because most of us are not privy to the terminology. Maybe as an introduction to metastable epiallias, maybe you can start talking about the agouti model, because I think that's a good example.
Right.
Give us a headway.
So I'm going to right now. So Benny, when I get on here and share, I might ask you to how to unshare again,
Yep,
but I'm going to
no
try
problem.
this.
Yep.
So this is the I call these the agouti sisters, and it's this experiment that our laboratory is most known for. From our standpoint, this was a very simple experiment, not to really look at metastable epiallials. And actually what we demonstrated ultimately was the mechanisms for what's called the fetal origins of adult disease susceptibility, which in the old days was called the Barker hypothesis. We use this model system to address this a simple question, and that is, are epigenetically regulated regulatory elements, can these be altered by environmental factors? That was the question. Very simple. So these are two mice. They're both female. They're both 100 days old. And interestingly and importantly, because they're inbred, they're basically genetically identical. Yet they look phenotypically massively different. And the only difference between the yellow animal and the brown animal on the right is what the mother ate when they were in utero. So the mother on the offspring on the left, the mother ate normal mouse chow, and the mouse on the offspring on the right, the brown one, its mother ate mouse chow that was supplemented with methyl donors. Why methyl donors? Because the markings and programming of the genome is dependent, one system is dependent upon putting methyl groups on cytosines when there's CG dinucleotides, 5' and 3'. And that's the marking system for, and usually when something's methylated, the chromatin condenses and the gene's turned off. Now, there's also histone marks because the DNA is wrapped around histones, and these work in tandem basically now to control the expression of genes. So this is the programming system. So rather than ones and zeros like we use them when we program a computer, here you're using chemical marks to program the expression of genes that gives rise to, as I say, these 260 different cell types and other kinds, if they're deregulated, other kinds of disorders and stuff. And so all the methyl groups come in from your diet. So what we did then, this is Rob Waterland, with this model system, and there's a transposable element that's upstream of the agouti gene. When that's completely methylated, then the gene goes back to normal regulation, developmental regulation, and you get the brown animal. And then why is it brown? Because the hair shaft is black, and right at the end of the development of the hair follicle, the agouti gene normally is just turned on. It puts a yellow band at the base of the hair shaft, and we now see the animal not as a black animal, but as an agouti animal, a brown animal. And that merges and melds into the environment a lot better than a black animal, and surely a lot better than a yellow animal would. In the obese animal, that transposable element that's upstream of the agouti gene is unmethylated early on. The decision was made to not methylate it. So the agouti gene now is inappropriately, there's another start site in that transposable element. And as a consequence, the agouti gene is inappropriately turned on throughout the whole animal, throughout its whole life. That means that that yellow, which should only be at the base of the hair follicle, is now the whole color of the hair follicle, and that's why the animal is golden yellow. The reason why this animal becomes obese, gets diabetes and cancer, always when they're yellow, is because the agouti gene is inappropriately expressed in the brain, in the satiation center of the brain, and binds to the melanocortin 4 receptor. So the animal never knows that it's full, and it's always hungry. So as a consequence, it literally eats itself into obesity, diabetes, and cancer. So now you have a model system where you have not only coat color being regulated epigenetically, but also disease susceptibility being regulated epigenetically, and it's regulated completely by epigenetic phenomenon. So it now becomes a beautiful in vivo model system for studying the effects of external factors on the development of diseases. So the first thing we did, since Rob was a clinician, not a clinician, not a clinician, he's a PhD, but he's a nutritionist. He wanted to look at the role of nutrition on the ability to determine the coat color of these animals, with the thought if you put enough methyl donors into the system that you could shift the distribution of offspring from being about half yellow and half brown to being almost all brown, let's say. And so we set up this model, we set up this study, and lo and behold, that's what he showed very, very clearly. And not only that, but then demonstrated clearly for the first time that this was completely determined by the epigenetic methylation of the transposable element that's upstream of the gene. So now you have epigenetics for the first time linked to disease susceptibility in a model system. And since these were offspring, this became, or is basically now, and people realize this. At that time, we realized it, but it took a while for other people to accept it, is the mechanism for what was referred to as the fetal origins of adult disease susceptibility that was first proposed by Barker back many years ago as an epidemiologist from the data that came out of the Dutch famine, which were humans that were exposed to severe starvation. And some of the women that were during the World War II in Holland, when Hitler put a embargo basically on food coming into Holland at the end of the World War II. And there were women that got pregnant, and they found out then later on, the epidemiologists, if the women were pregnant and the babies were in utero during this period of starvation, they had higher incidences of obesity, diabetes, cardiovascular effects, and a doubling of the behavioral phenomena of schizophrenia. So low doses or low amounts of food increase the incidence of schizophrenia in humans. But the mechanism for this was not known. So as you can imagine, Benny, because you've experienced this with the transgenerational people programs and stuff that you've done and studies that you've done, people are skeptics and hugely skeptical, and they basically swept all that stuff under the table. They couldn't understand how that could possibly occur, because we all know that, I'm using this in quotes now, we all know that diseases is a genetic disease, a genetic problem. If it's a genetic problem, it can't work. There's no memory system. But when you bring in the epigenome, which is what's causing it, it's the mechanism for this. Now this can happen, and there is now a mechanism for it, and the whole field of the fetal origins of adult disease susceptibility has just blossomed, because people now are not just looking at genetic mutations, they're also looking at changes in the epigenome. And together, when you look at both of these together, you finally get a much better and fuller picture of the mechanisms that are causing disease susceptibility in us and in all mammals.
Thank you for the explanation, Randy. Just to clarify, methyl donors, the methylation that Randy's talking about is one carbon metabolism primarily, is the folate pathway. And so it's folate, it's choline, it's those types of agents. And that's important to know. And the main issue here is that you are demonstrating that the environment can modify the expression of genes for generations. And that's really the key. And Randy, maybe you can talk a little bit. I know that's not your work in particular, but you talked about nutrition, how it affects the genes. But I know you're aware of behavioral modifications and behavior of the mother with her young offspring that would modify their disease susceptibility later in life. Could you tell us a little bit about that?
Yeah, this is work by Michael Meany and Moshe Schiff. Our paper was on, so what we did, what Rob Waterl and I did was with nutrition was in utero. When all the marks are being placed on to basically to give rise to the whole fetus and ultimately the individual or offspring that are born, they're all laid down in that really early period of time. So that's an extremely vulnerable time for other environmental factors to come in and usually mess this system up. But the other question is, can the epigenome be changed after birth, postnatally? And potentially, can it be changed by things as subtle as maternal behavior? I mean, this is really, it's truly astonishing, actually, but it's true. So 2003, we published our paper on the Agouti gene study. 2004, Moshe Schiff and Michael Meany studied what I call the licking rat experiment, where you had animals that were mothers that were lickers and groomers, and then you had other, these are just normal in the population of outbred rats. Then you had other mothers that basically were hands off, as they would say. In other words, they hardly touched them at all. And the offspring then of those hands off, for example, mothers became hands off themselves. And they're very hyper animals. I mean, you hit the cage and they'll pop right out of the cage. Whereas the other ones, they're the lickers and groomers, the mothers now, I don't know what happens to the fathers. We're focusing on the mothers now. They become lickers and groomers also. And they're very laid back. You hit the cage and they say, "What's going on?" That kind of stuff. So it's very, very different behaviors and very different. And it looks almost like it's genetically inherited, but that's not what was happening here. They found out that the physical tactile touching of the offspring, the licking and grooming, in the brain caused the release of many, changed the expression of many genes, but the glucocorticoid receptor, for example, that's involved in stress responses was clear. The licking and grooming caused the release of methyl marks in the promoter region of the glucocorticoid receptor. Whereas the hands off, the mark stayed and they were hyper all their life. So it was a behavioral effect that was affecting the epigenome that was now affecting the behavior of the offspring. It was a truly astonishing study. And then the next one in 2005, Benny, which you're propagating farther and forward, is that these marks are not always even completely released when they go through the egg and the sperm. And there's evidence now, including the results of your studies, that show that there's a transgenerational effect, that these can be epigenetic marks can be carried forward transgenerationally. And the first study that was published on that was Michael Skinner's work in 2005. So 2003, 2004, 2005, in effect now, the whole field of environmental epigenomics, the little squares on the jigsaw puzzle around, that was completely defined. Now what are we doing? We're filling in this massive jigsaw puzzle, the pieces inside, but we now know that the epigenome plays a huge role in disease susceptibility. Before that, that was not known, or if it was known, people didn't really pay much attention to it. Totally changed.
Thank you. I'd like to have people ask questions if they can, but I'll make two comments first. One, you may or may not know this, but Kirk Hogan and I used the agouti mouse a few years ago and gave the mothers nitrous oxide, which is commonly used anesthetic, and we significantly changed the coat color of the offspring.
In the yellow way, right?
That's right. That's right. So that was a debilitating, a negative effect on the offspring by using an anesthetic. So this is also opening up a whole new field of perioperative
Right.
changes. What are we doing when we operate on patients? What are we doing when we anesthetize them? When we're cutting into them, really? And Kirk may want to add more to this in a minute. But the next thing that would be nice to talk about later is we're not just talking about disease susceptibility, but we're talking about uh, developmental susceptibility. So there are susceptible periods for epigenetic change, right? So embryonic stages, like you
Right.
mentioned, puberty,
Right.
and I assume old age.
Because
Maybe,
everything is falling apart,
yeah. So maybe at
including the
some point,
epigenome.
yeah, yeah. So maybe you can talk to us a little bit about this, but let's see if there are others who have questions first, so that I don't fill the whole time.
Well, could I just ask about the needle in the haystack principle? There are so many potential targets in this language that you're defining. How do you choose this might be the important one that I want to study? That's important for any scientist to know in any field.
That's a very good question, and it's even more complex than maybe even that what you realize, because epidemiologists can usually only get samples from blood, buccal swabs, etc. So if you're interested, for example, and this is just an example, but it's an important one, and I think it'll answer your question at least in part. Uh, if you're interested in, you know, autism, schizophrenia, psychosis, these kinds of things, psychopathy, any of these types of behavioral issues, you're not going to be able to drill into someone's head and get a sample from that and look at it epigenetically. Now, you might be lucky enough to have quick autopsy so that you could get brain tissues and look at after people die and that type of thing. But it doesn't provide for a very good way of doing the kinds of things you're talking about, sort of robust epidemiological studies with genes that are known to be problematic and cause disease susceptibilities, and ones that you would be interested in looking at, okay? That's what you're kind of asking, I think. So here is what we did, and we started out with this, and maybe I didn't make this as clear as I should. We then decided that we wanted to determine in humans the complete human imprintome. Why? Because these imprint regulatory elements are established in the gametes. They're the inherited marks. That's why you get parent of origin dependent expression. Or they can be established off of those right after fertilization in the early embryonic stem cells before there's any development that has occurred. And these genes we know are incredibly important in behavioral determination, development of the fetus, and also in cancer formation. So, and remember, these marks established before you had differentiated cell types. So now, by looking at those and asking questions, what exposures, particularly in utero, or maybe in adulthood, but mainly in utero, very early, which is what pediatrics is all about, the early stages. You're the people that sit at the front. You're right at the heart of all of this stuff, where getting samples and stuff is incredibly important for addressing these early events that give rise to developmental disorders like schizophrenia and autism. These are developmental disorders that have started way back when, probably at the very earliest stages of development. And my guess is that many of the genes that are deregulated in autism and schizophrenia are going to be shown, and some of them have already, to be imprinted. So now the question is, what environmental factors are potentially altering these imprint marks? And the beauty of using imprint regulatory elements for these studies in adulthood is because the marks were made so early that you can take any cell type in your body and you will be able to monitor that change in that individual. And then you have a phenotype because you know the person has schizophrenia, and you look for the genes that are involved in schizophrenia that are imprinted and ask simple questions. Are the imprint regulatory elements altered? And if so, which ones? This has not been done, but I'm telling you, it's unbelievably powerful because just what I was telling you about, like Down syndrome and DeGeorge syndrome, there's schizophrenia that occurs in these people and autism. Nobody before this knew. You're the first people that know this. There are 4 imprint regulatory elements in that domain. How hard is it now in DeGeorge syndrome people to assess the imprint status of those regions? It's not hard at all, but it's damn hard when you didn't have any clue that they were there.
Yep.
I'm an emotional person, but I mean, we're in the middle of writing this, and I am actually astounded myself, and it's hard to astound me. It's incredible. You can take any disease disorder that you want now and screen for the role of imprinting in its etiology. And the same thing is true now with Rob's map for these crazy metastable epialleles, because again, that mark is established. It's like in that agouti mice that you're looked at so early in development that you can actually screen for these changes in adulthood in any cell type in the body.
Randy, this is Kirk. Could I ask a question that I always get asked? And that is there any trait that's not epigenetic?
My guess is probably not. It's going to be genetic. So how do we find some of these things? We find them like KCNK9. It's an imprinted gene that's involved in breast cancer formation. I'm working on this paper right now too, so it's near and dear to my heart. I mean, normally it's overexpressed and it's involved in breast cancer formation, and it's basically amplified genetically, right? So everybody, that's the way. But now we know, because this is what we found, it's also amplified its expression through loss of imprinting. So my point is that my guess is that anything that we have as a disease or a disorder is going to have a genetic component and an epigenetic component. And that is particularly true if imprinted genes and metastable epialleles are involved in their etiology. That's my guess, Kurt.
Kirk, since you spoke, maybe you can give our audience a small spiel on anesthesia and epigenetics and what we're not doing right in the OR. Sorry to put you on the spot.
First point is we're focusing on diseases and disorders, but I would argue that beneficial traits are also all epigenetically inflected, like perfect pitch or being ambidextrous or whatever else you might think. When I answer the question, there's literally no trait that doesn't have epigenetic influences, because that's how the whole thing is coordinated. As far as surgery and anesthesia goes, these are incredible interventions that we did not evolve for. And when you stop to think about putting someone into an unconscious state and strapping them down and paralyzing them, and then sawing on their bones, opening their skulls or whatever, you would have to think that there would be changes in the epigenome that arise from that have consequences after surgery. And so the first trickling amounts of data are coming out, and they're really astounding. And I guess the major point about this is that the epigenome, and DNA methylation in particular, has a tempo that we're just beginning to understand. It doesn't happen, it does happen over years and months, but we're seeing evidence with Reed Alish and others that the epigenome changes in a matter of hours and days after environmental exposures. And these have consequences on the phenotypes and the traits that is just a wide open universe. So like Randy conveys, I would say we're at the point now where we got our first telescopes to look up at the night sky. There's literally nowhere you can point that you won't see something for the first time. And since I'm not aware of anything as incredible as anesthesia and surgery, that's a big focus of what we're doing. But we're also looking at COVID. We're also looking at Alzheimer's disease. We're looking at cognitive aging among women, differences between rural and urban and suburban childhoods on DNA methylation patterns. We're seeing changes in every one of these things. And my final point is these are useful for susceptibility and risk and prognosis assessment, but they're also modifiable. In other words, whether the genes are turned on and off is something that we have as a whole new world of therapeutic targets.
Yeah, and the thing is, I like that analogy with the telescope, Kirk, because in a way, I wasn't there when Galileo looked at Jupiter and saw moons revolving around it, but it must have been in a feeling of just huge excitement. And that's what I feel when we're using the imprintome and looking at various diseases. And my goodness, it's incredible. It actually is incredible.
Yeah, the other
And
amazing
stuff
thing
that
is
nobody ever saw before.
the amazing thing is the platforms are so good. Our methylome sequencing technologies
Yep.
and everything, they're drop dead. The rate limiting step in beginning to exploit, like John Wolfe used to say, it's like walking into the golden throne room with the dragon in Lord of the Rings with a little flashlight. Everywhere you look, you see something. The rate limiting step is to train more graduate students and postgraduate students like Colin and Andy and Reed's lab and others. We need hundreds, if not thousands of people to really figure out what this universe looks like.
Yeah, I mean, this room that we've kicked open, I mean, it is so darn huge. It's huge. It's huge. I tell people, I said, you don't have to worry about competing with other people. There is so darn much stuff in here that you can just pick up gold nuggets, every one of them. There's enough gold nuggets to go around.
I guess another point is that's encouraging that the NIH, at least some of the study sections, are beginning to share this point of view.
Well, I tell you, this is something Benny said in the beginning. It sure the hell wasn't like that in the beginning. I want to say something now about this, Kurt, because everything that we've done, because we even did the whole thing about the evolution of imprinting. We showed that imprinting evolved 150 million years ago in a common ancestor to marsupials and eutherians, and so it occurred with the advent of placentation. That study was not supported by NIH. This was supported by private funds. And the same thing actually with the agouti mouse study. Now, we got plenty of money after we got these things done, but the agouti mouse study, except for Rob's salary, which came from Dan and Yogurt, but the rest of it was private funds that had come into the laboratory. If you would have proposed, or if I would have proposed either one of those studies to NIH, I can guarantee with 100% accuracy that it would not have been funded. And yet, this is what we're most known for.
Maybe the two of you and maybe Reed can comment on the role of clinicians in all of this. I don't think the clinician's role is only to provide tissues. We have a perspective that the scientific community doesn't necessarily have. We come from a different angle. Could you maybe, the three of you, comment on how you marry these two?
Well, I'll jump in on that. And that is, what is autism? What is schizophrenia? What is asthma? If you pick up a book and a journal article and read it, you'll think you understand it, but you have to have boots in the trenches to understand what these phenotypes are. Alzheimer's disease is not a disease. At best, it's a spectrum, probably more of a syndrome. At the tail ends, it's shading off into Lewy body and other forms of dementia and overlapping. And to really know what your trait is that you're going to map on the methylome, you have to have boots on the ground, years of experience, understanding of how we measure these phenotypes and categorize them, because otherwise your wonderful platforms will just crash and burn.
Yeah, and I mean, it's interesting because I'm going to be a little more negative because I came out of into biology through radiation biology, and there always historically was a strong connection between the clinicians, the radiation oncologists, and radiation biology people for years and years and years. At the end of my career, that really had eroded to the point where, in effect, the clinicians had to spend most of their time treating people and, you know, making money for their salaries in effect, so that there was very little time left, Benny, for the clinicians to really be involved in sort of basic research, which was always the case in radiation oncology. Then you go to the field that you guys are in, you know, where it's neurosurgery. I would imagine that's even more difficult, because the pressures to, you know, to treat people, and which are real, are huge, and to get time to do research and get groups together and that can do research with PhDs and other MDs is very, very difficult. I think you and Kurt are probably you're the abnormal, not the normal. That's my guess.
I'm sure Bob
I
Dempsey
mean
has some
abnormal
thoughts about this.
personally. I mean that it's unusual that people like yourself are involved in this at this level of basic research. It's
But Randy, it requires huge support from the department and institution, because we cannot be applying for 5 grants or 10
right.
grants per year. We can't. So it really requires a commitment from the department and the institution. Not many institutions provide that.
That's
We are
right.
provided this here.
Yeah,
It's getting less and less.
it's been something that really is the hallmark of this department. And I think 45 applications went out during COVID by this group of very overworked surgeons
Yeah.
and a couple of scientists. So it's been a volitional desire, and it attracted these great people here.
And the thing is, as I said, you know, the pediatrics, this is where the game begins. You guys are at ground zero, as they would say. And it's so exciting, because even though Benny said more than getting tissues is a huge issue. And it's very, very important in this kind of research now. Once you have the targets like we defined them, but if you don't have tissues to look at, you can't do anything.
Yeah, and we do, and we have to think that way. I do atherosclerotic patients, and I have living atherosclerotic tissue, and we found 247 differentially regulated genes when they become symptomatic. And we are trying to figure all that out, and that's just busy clinicians.
Right.
Right.
So,
Great.
Randy, some of our surgeons are going to start to wean off here to go to the operating room, but we'll be staying on as long as you want to stay on. Maybe you can tell us before time escapes us, what it's like swimming against the waves. How did you stick to your guns when you weren't getting funded for this type of research? When I don't know how supportive Duke was. I imagine that there were some tough moments there. What is your advice to people who are going against the grain, so to speak?
I
Or
mean,
am I wrong? Maybe it was smooth sailing for you.
no, it was rough. Um, as I said, I was fortunate enough to have been interacting, because people in the toxicology field realized that there was a whole group of toxicants that are non-genotoxic. Well, these non-genotoxic compounds were causing cancer. Well, now we know they're causing cancer through alterations primarily in the epigenome, not genetic mutations. But back then we didn't really know that, but they were interested in the work we were doing along those areas. And initially, as I said, it was in genomic imprinting. And I got resources from a number of pharmaceutical companies with no strings attached that I could put into an account, and it enabled me to do the studies that I was telling you about. So having money that's not NIH dependent is a real plus in doing not crazy experiments, but studies that are a little off the normal beaten track. I was fortunate enough, and maybe not fortunate, because I have somewhat of a gift of the gab, so I was able to get people interested and get them to support it. But that's one way to do this, because if you're dependent only upon NIH, it's going to be problematic doing a lot of, I think, uh, creative type studies. They're good at supporting things when you have to fill in the blanks, and there's a lot of filling in the blanks right now. But starting new fields and doing groundbreaking stuff. Now they're not particularly good at that, even though they say, yeah, we're going to do that. They never do that. Why? Because the reviewers basically are normal people, and they want to have a good probability that they're going to have success. Well, when you're doing things like you guys were doing and we were doing, there's hardly any probability of success other than you've got a good feeling that there's going to be success, but you don't get the money. It's hard. It's really, really hard. And then, as I said, at the end of my career, the department support and stuff started falling away because the clinician's role that was played in research became less and less and less. And also people found it more and more difficult to understand how epigenetics could possibly be involved in what they were doing, which was treating patients with large doses of ionizing radiation. So there's became kind of a mismatch that spread apart, and that became ultimately became problematic, and really in a way was the reason why I ultimately retired. As you know, I didn't retire. I went over to NC State because my group went over there, and now I function more as a consultant, but I didn't get my fingers out of the mix. I retired from running a laboratory directly. Is what I did. I don't know how to tell people, Benny, you know more about it yourself. When you get beat on the head, our papers were turned down, and just like, just like you incurred experience, it's awful. And even worse than that, you worry that you won't get any support. If you didn't have any backup money, as I said, I don't know. It's really tough. I think a lot of people just don't do it because you get beat on the head long enough, you're going to say, I've had enough of this fun. It's tough.
Yeah. Yeah, Kirk and I are keeping track of all the rejections, and we're going to make a poster at the end of it.
You'll have a big one.
Yeah. Any questions from anyone else about epigenetics, about life?
But on the positive side, when things work, Benny, when
It's
you get
amazing.
things that make sense, it's unbelievably exciting, more exciting than you could imagine, really.
Yeah. Maybe briefly tell us about how epigenetics will change our understanding of clinical disease. I mean, you've been talking about this, but is there something more obvious for clinicians to know?
Well, I think people are starting to do this now. I mean, now that we know that people realize that the epigenome is also potentially involved in disease formation, you can start using that as another area of treatment for therapy.
Yeah.
And
in fact, that started.
Yeah,
For example, the biggest, the most deadly tumor that we care for in pediatric neurosurgery is the diffuse intrinsic pontine glioma, and it was discovered a few years ago that the one marker that's pathognomonic for this tumor is a mutation in one of the histones, H3K27M. So directing epigenetic therapy to this is going to be the way in the future, not surgery, not traditional chemotherapy.
right.
Yeah.
Yeah, so I mean it's just another way of looking at therapy then.
Yeah.
So that's one aspect of it. But I don't have anything much else to say about it. I mean, I've always been interested in having things clinically used clinically, the information, but I've obviously been more interested in, I'm sort of an explorer. My approach is once things have been defined, I go off and I do something else. I don't use the agouti mouse model now, but from the standpoint of toxicological studies and stuff, it's a very powerful tool, and a lot of people, I'm sure, are using it now. But it's not something that I'm interested in now. I was always interested, I said, in determining human imprintome. That's done now. At least version 1.0 is done. And that's going to provide us with, and other people, more information than you can imagine. I mean, it's going to be a huge pipe opening up and a whole nother way of looking at these diseases. And maybe as again, now with CRISPR and the ability to maybe go in and change not only mutations, but even epigenetic regulatory elements, it could be a mechanism, a way you can't treat something unless you have a better idea of how it's happening genetically and epigenetically. So that's what we provide.
So for the very last minute, thanks to Kirk Hogan and Bob Dempsey, Reed Eilish became part of our department 3 years
Right.
ago, and he's our primary epigeneticist now, and we're forging a lot together with other members of faculty. Maybe, Reed, you can close it up and either make a comment or ask a question of Randy.
Yeah, sure. I'd like to. Thank you, Randy. This has been really, really enlightening, even for somebody who's been focused on this for a long time, like myself. One question I have for you is about the timing of detection. Like you're talking about all these epialleles and these changes that we're finding associated with different syndromes, such as Down syndrome. But what do you envision that we're going to need to do in terms of the timing of the detection? We can't really, in most cases, probably wait until the phenotype is already present. So
Right.
is there going to be things that we're going to need to do, sort of right at birth, maybe, or what are your comments on that? Like in terms of how are we going to identify these changes or problems, if you will, early enough to actually have meaningful outcomes?
Yeah, that is an interesting question. As I said, I keep focusing on imprinting because when we find alterations in the regulatory elements, epigenetic alterations, we know that those alterations occurred very early. So we're talking about a developmental problem, basically, that's occurring. So now let's say there's genes that are involved in autism, schizophrenia that are unmethylated or methylated, and we find these people. Then the question is, how the heck do you go in and potentially change, what do you do at that point? That we're not there yet. So you're talking about detection. You can screen people at any point for these changes. And I don't think it's going to take a lot of time now that we've got, at least with imprinting, to define the genes that are playing huge roles in many of the major behavioral disorders and even cancers. Because if you think about it, with cancer, these changes have to, when they occur early, most of them probably are going to block apoptosis somehow so the cell can live as it accumulates more and more garbage mutations. I don't know. I mean, even if you knew this, Reed, that this gene, let's say gene X, is epigenetically changed, you'd have to have a mechanism by which to go in and alter it. But you at least would, if you had potential ways of doing that, you could now at least monitor it. That's possible because you know where you're looking. What do you think? I don't know. I mean, we're not there yet, I don't think, are we?
No, I don't think we are. But I think I think your point is well taken, that at least we know where to look, and then we can sort of start to work backwards from there. And as you said, manage it, monitor it, and eventually we get to the point where we can modify it towards
Right.
healthy outcomes.
Now, the point I want to make too is, in closing here, I focus on imprinted genes and metastable epialleles because they're regulated epigenetically and the marks are placed so early that you can literally monitor these from any cell type in the body. And when you find a change, you know the change occurred very early. That doesn't mean that there aren't epigenetic changes that occur later and that ones that are important. But when you start in a field like this, that's so big, you've got to get it down to areas that you know are really important and ones that you can readily, easily monitor. And that's why we went with the imprint regulatory elements and Rob went with the metastable epialleles. I'm sure of it, because we wanted model systems in humans that are somewhat comparable to that wonderful, beautiful agouti mouse system that we first started the whole field of environmental epigenomics with. And that's why we went with imprinted genes and Rob went with metastable epialleles.
Randy, thank you so much. This was amazing, and hopefully it'll encourage more conversations between clinicians and scientists, and I hope you enjoyed it as well.
I did. Did you like the interview thing? It worked well, didn't it, Benny?
Many thanks, Randy. This is great, and it was great for us and our different levels. So that's
Right.
wonderful. Thank you again.
You're welcome. Thanks.
Thank
Keep up the good work.
All right. Bye-bye.